Hello, students. Today, our topic is physiology of bacteria, growth and reproduction of bacteria. You know the structure and the morphology of microorganisms, their cell, and now we'll study how does it work. Why this topic is so important for medical students? The knowledge of physiology of bacteria is necessary to perform the bacteriological diagnosis of infection disease. It means the isolation of bacterial culture and special nutrition media, and then it's the identification of microorganisms according to their morphological, biochemical, and other properties. It is this method, bacteriological, is the gold standard, the most reliable, very sensitive and specific method in diagnosis of almost all bacterial diseases. And here you see the schedule to the lecture. We start with the chemical composition of bacteria, then come to the types of microbial nutrition, and we will start with the bacterial transport system. The central question of our lecture is types of respiration in microorganisms, and also we'll study the growth and reproduction of microorganisms and nutrition media used for their cultivation in microbiological laboratory. In chemical composition of bacteria, the water consists with 70 to 90%, and other 10 to 30% is a dry weight. And this dry weight includes organic and inorganic components. Among the dry components of bacterial cells, the proteins is the first position, more than one half of dry weight, and the proteins play a role of enzymes, the biological catalysts, and also they participate in building of all cellular organella. At the second position, you see the RNA, 20% of the dry weight, and it is higher than in eukaryotic cell. And you should remember that there are three types of RNA in each live cell. It's the messenger RNA, transport RNA, and ribosomal, and all of them involved into synthesis of proteins. The DNA contained in bacteria is approximately 3%, and they have only one molecule, only one chromosome, the double strand circular DNA, and it's not separated from the cytoplasm. The lipids, it's mostly phospholipids, the component of cellular membrane, and some microorganisms may contain the lipids as inclusions. And this substance, lipopolysaccharides, are present only in gram-negative bacteria in the composition of their outer membrane. And peptidoglycan, a very specific substance which find in gram-positive and gram-negative bacteria but in different concentration. And it's only one molecule, but giant molecule surrounding all the bacterial cells. And the microorganisms take the nutrients from the environment to build all these components of the cell and to get the energy. And what may be nutrient for bacteria? Uh, Almost all substances, starting with human food or to some inorganic compounds. And bacteria have very diverse types of metabolism. And what does it mean, microbial metabolism? The microbial cell, like all other life cell, is the open system. It means that it can live only um, in case of constant exchange of energy and substances between bacterial cell and environment. If this exchange stops, the bacteria die. And this exchange of energy and substances, this metabolism, consists of two processes, catabolism or dissimilation and anabolism or assimilation. These two processes took place at the same bacterial cell and at the same time, but they have different direction. In catabolic reaction and dissimilation, bacteria take some substances from environment and decompose it inside the cell to produce the energy, and this energy is necessary for biosynthesis, for bacterial movement, for growth, for all function of bacterial cell. In a reaction of assimilation or anabolism, bacteria take from environment the foreign substances and transform them into the component of own cell, into own proteins, carbohydrates, nucleic acid, and other necessary substances. They microorganisms have different energy requirements. One of them can accumulate the energy of sunlight, and these microorganisms, named phototrophs, they can transform physical energy of some radiation, into chemical into energy of ATP. And this phototrophic bacteria is non-pathogenic, mostly free-living um, microorganisms. Other microorganisms use the energy of chemical reactions, oxidative reductive reactions. And these microorganisms named phototrophs, excuse me, chemotrophs. And they divided into two groups according the substance which they oxidize. If they oxidize inorganic compounds like H2, H2S, iron, ammonia, or others, they named chemolithotrophs, and they transform chemical energy into chemical and produce ATP. And these microorganisms, also non-pathogenic, mostly free-living soil bacteria. And the other chemotrophs take their energy oxidizing organic compounds like glucose or other carbohydrates, organic acids or amino acids and others, and they named chemoorganotrophs. They decompose these organics to produce the energy, and they also transform chemical energy into chemical energy of ATP. Microorganisms are also different in their metabolic requirements, especially in carbon source. You know that all organic compounds are the compounds of carbon. So this classification according to the carbon source is very important. Um, the first group of microorganisms can use the inorganic source of carbon in form of CO2, carbon dioxide, and these microorganisms named autotrophs. They use inorganic carbon to produce organic matters. Some of them photosynthetic, they use energy of sunlight and assimilate the CO2 and produce um, photosynthesis. And other use energy of chemical reactions and also assimilate CO2. Um, and all these microorganisms are non-pathogenic for people. Heterotrophs need some organic pre-source of carbon. It may be glucose or some other organic matters, and they transform one type of organics into other. And pathogenic and occasionally pathogenic bacteria belong to this group, the heterotrophs. And more details about bacteria with different type of metabolism. Photosynthetic autotrophic bacteria, you see example here, as the purple sulfur bacteria. They contain the purple pigment to assimilate the sunlight, and they're non-pathogenic, of course. They live in hot springs in temperature of 70 degrees Celsius in high concentration of salts, and they use the salts to produce organic compounds and then assimilate CO2 from the air. Chemosynthetic, chemolithotropic bacteria mostly live in soil, and they are very important in the circulation of elements in the biosphere, um, participate in cycles of carbon and nitrogen and some other elements, and all of them are non-pathogenic. Heterotrophic bacteria divided into two groups, saprophytic and parasitic. Saprophytic use dead organic matters. They mostly live in soil, and they produce decay of some biological waste, the animal carcasses, and dead plants, and they produce the mineralization of organics. And the saprophytic Sulfuric bacteria are mostly non-pathogenic, but sometimes they can be occasionally pathogenic. It means that they cannot cause disease in healthy people, but can cause harm to person with a weak immune system. And the parasitic bacteria take their nutrients from the live host. And of course, they are pathogenic. They can cause disease. And some example you see here of this bacteria is the Bacillus anthracis with very specific morphology, the gram-positive large rods arranged into chains, so they are streptobacilli. And they infect the skin and subcutaneous tissues and produce a specific lesion with black scar, is characteristic for anthrax. And in our classes, we will study mostly these parasitic bacteria, which are pathogenic. Mhm. And at this slide, you see the summary about possible types of microbial metabolism. But what is most important for us that all pathogenic bacteria are chemo, organo, heterotrophs in their metabolism. It means that they use energy of chemical reactions and then oxidize organic matters to produce energy. And for biosynthesis, also need the organic compounds. And students, you understand that we have the same metabolism. Humans are also chemo-organo heterotrophs like pathogenic bacteria. And here you see the classification of microorganisms according to their requirement in nitrogen. From biochemistry, you remember that among organic compounds, some are not nitrogen containing like lipids and carbohydrates, but others like proteins and nucleic acids, they're nitrogen containing and they're essential for bacterial life and growth. Some microorganisms can assimilate the molecular nitrogen from the atmosphere and in two form. It is process named nitrogen fixation, and some of them can use this inorganic compounds of nitrogen, like nitrates or nitrate ion. Mostly they are soil bacteria, some of them free living and autotrophic, and others live in symbiosis with plants. And this is mutual benefits for both participants of the symbiosis. The bacteria provide the plants with organic nitrogen, and instead the plant provides them with glucose, like organic source of carbon. This bacteria is non-pathogenic, but they're very important in agriculture. Many microorganisms can assimilate the nitrogen in the form of ammonia because it's a part of amino acids. This amino group present in all amino acids, the blocks of building blocks of proteins. If bacteria belong to this group, it named a prototroph. It means that they have very simple nutrition requirement. The prototrophs can grow in simple nutrition media like meat peptone broths, meat peptone agar, and moreover, they can grow in minimal nutrition media. Minimal nutrition media means that we take the distilled water, add only one substance containing organic carbon. It may be glucose or even citric acid, and we add the ammonia salts, the inorganic source of phosphorus, sulfur, calcium, kalium, iron, and other elements. And prototrophic bacteria can use this solution to synthesize all necessary proteins, nucleic acids, lipids, carbohydrates, vitamins, all they know. Mostly prototrophs are saprophytic bacteria, but they can be symbiont of human body, like E. coli, you see it in this slide, intestinal bacteria. And sometimes they are occasionally pathogenic, can cause disease in immunocompromised person. And the third group of microorganisms in the source of nitrogen, need the organic compounds with nitrogen. It may be amino acids or nucleotides or some vitamins. The bacteria need them but cannot synthesize by themselves, and they must take it from the nutrition media. These bacteria named auxotrophs, it means they need some auxin, some substance like amino acid or vitamin for their growth. And this substance auxin the same as a growth factor. If this growth factor present in nutrition media, bacteria can grow. If this substance absent, bacteria cannot grow. So it's a growth factor for them. The cultivation of auxotrophic bacteria is more difficult than prototrophs. They cannot grow in minimal nutrition media, cannot grow in simple nutrition media, and we use for them media with animal tissue, the blood agar, or agar with added brain tissue, or liver tissue, egg yolk, or yeast extracts, and some other substances. And the pathogenic bacteria often have properties of auxotrophs because they live inside human body, and in this environment they're surrounded with all necessary organic compounds, and it's not essential for them to synthesize it. So these bacteria lose the ability to produce some amino acid or some vitamins, and instead they acquire the factors of pathogenicity, some properties to cause harm spread in human body. And in this slide, you see the example of oxotrophic bacteria. It's a Neisseria meningitis, which causes the infection of nervous system in people, cerebrospinal epidemic meningitis. It's diplococci, it's a gram-negative, very typical morphology and difficult to cultivate them in laboratory. And how bacteria take the necessary nutrients from the environment. Students remember that no any phagocytosis in bacteria. Their cell is too small and it's surrounded with a rigid cell wall, and they cannot produce pseudopodia or something like this to engulf the food particles. They can only absorb the soluble nutrients through all their surface. And, due to this rigid cell wall, bacteria can ingest only small molecules, and the large biopolymers like proteins or polysaccharides cannot be transported through this cell wall and through the membrane. Firstly, bacteria produce the hydrolytic enzyme and release it outside of the cell. And this enzyme cleaves the large molecules into smaller fragments, like uh, large protein molecule into polypeptides, or large molecule of polysaccharide to monosaccharides or oligosaccharides. And then these small molecules can be absorbed inside bacterial cell. And bacteria have special transport system to take nutrients and water and salts from the environment. It's possible the passive transport, which doesn't require the energy of ATP, and it's possible only along the concentration gradient, when the concentration of some substance high outside the bacterial cell and low inside. And along this gradient, in the process of simple diffusion, the small molecules can be transmitted through membrane, through this phospholipid bilayer. This process is not biological, it's just physical, chemical, and cellular membrane serves only like a semi-permeable barrier between cytoplasm and environment. More complicated passive transport is facilitated diffusion, and you see the difference. In this case, the special proteins, porins, participate in it. They insert it into phospholipid bilayer and produce the spores. And of course, it's easy for small molecules to come inside the bacterial cell then through the um, phospholipid bilayer. And this process also possible only along the concentration gradient and doesn't require energy of ATP. Bacteria can take their food from environment if some nutrients present outside in low concentration and the concentration inside is high. And so it's possible to transport against the gradient concentration. Of course, it requires energy of ATP and participation of special transport systems, proteins named permeases. These permeases incorporated into phospholipid bilayer, and firstly they take this position. The active center is the outer side of membrane, and it reacts with transported substrate. Then this molecule turn into 180 degrees and bring this molecule inside the bacterial cell and release it into cytoplasm and then return to the previous position. Of course, these movements need the energy of ATP. And the fourth mechanism of transport in bacterial cell is the group translocation. Sometimes when some molecules transported through the cellular membrane with participation of protein permeases with energy of ATP, the structure of the molecule is transformed and these molecules easily involved into metabolic processes in the cytoplasm of bacterial cell. And now we come to the central question of our lecture is types of bacterial respiration and types of energy metabolism in microorganisms. There are two ways in which bacteria obtain their energy is the fermentation and respiration. In fermentation, the organic molecule like glucose split into two parts and one part oxidized as a expressed as a reduced, and as a result we have two molecules of lactic acid, also organic compound, and the small amount of energy released and saved in the ATP. In reaction of respiration, the same molecule, glucose, oxidized with O2 from air, and it decomposed for the end products like carbon dioxide and HO2. And this process allows to obtain more energy, which also saved in ATP. In some bacteria, they can take energy only in fermentation without oxygen, on using only these organics and producing some acids, lactic acid, or some other organic acids as a final product. Other microorganisms need oxygen for their respiration, but in overwhelming majority of microorganisms, they can easily switch the metabolism between these two processes, depending on the environmental condition. If these bacteria live in environment, in media, with high concentration of organics and absence of oxygen, they take energy in fermentation in this process. And if they live in media with low concentration of organics, but in presence of oxygen, they obtain the energy by respiration, producing this reaction. So the metabolism is reflexible and they have high adaptation for these different conditions. The bacteria differ in a type of respiration, and in the slide you see listed 6 different type of respiration of microorganisms. They may be obligate aerobes, microaerophils, obligate anaerobes, facultative anaerobes, the same as facultative aerobes, Some of anaerobes are aerotolerant, and very specific group is capnetic bacteria. And more details about each of this group. The aerobes, they need the oxygen for their growth and then can detoxify peroxides at the side products in biological oxidation. They detoxify peroxides using special enzymes like catalase and peroxidase. If we say obligate aerobes, so they cannot grow without oxygen. Among medical important bacteria, the obligate aerobes is here. It's the Vibrio cholera, it's a comma-shaped gram-negative microorganisms, causative agent of cholera, and the Mycobacterium tuberculosis as the acid-fast rod-shaped bacteria, which affect mostly lungs people, causing tuberculosis. Microaerophilic bacteria require oxygen, but in smaller amount that it's present in atmosphere. In atmosphere, the concentration of oxygen is 20%, but microaerophilic bacteria need 5 to 10%. The medical important microaerophils is the Helicobacter pylori. This is the curved bacteria, highly motile, gram-negative. They are causative agent of gastric ulcer and some other enteric diseases, very harmful bacteria. And lactobacilli, also macroaerophilic, but these microorganisms are very useful as they live in intestinal tract of people, and they can obtain the energy due to respiration, but mostly due to fermentation, and then produce a lot of lactic acid, and this lactic acid suppresses the growth of pathogenic enteric bacteria. So this lactobacilli protect us from Salmonella, Shigella, and other intestinal pathogens. Facultative anaerobes is a very large group of pathogenic and occasionally pathogenic bacteria. They can grow in presence of oxygen and in absence of oxygen. They can have enzymes to detoxify peroxides. And medical important facultative anaerobes you see here is the enteric bacteria like Escherichia, Salmonella, Shigella, Klebsiella, and it's gram-negative. And gram-positive Staphylococcus, Streptococcus, they're also facultative anaerobes. Obligate anaerobic bacteria, they don't utilize oxygen, and moreover, the oxygen can kill them. These bacteria have no enzymes to detoxify hydrogen peroxide, the toxic substance. You know that we use H2O2 like antiseptic in medicine. Due to metabolic process, the substance accumulates inside the cell and eventually kills them. The example of medical important obligate anaerobe is the Bacteroides, the gram-negative polymorphic bacteria, which can be found in intestinal tract of people, and also they can cause the inflammatory purulent process in abdomen and in chest, often in association with other microorganisms. They highly sensitive to the influence of oxygen, and they killed immediately in the contact with oxygen. And other anaerobes are rather aerotolerant. They don't utilize oxygen for their growth, but they can survive in presence of oxygen. Like Clostridia, you see in this slide, it's gram-positive large rods, and they're spore-forming. And in the contact with oxygen, the vegetative cells of Clostridia die, but spores survive because it's not metabolism in spores, they dormant form, no production of H2O2, and they stay alive in the presence of oxygen long time, many years. And the capnetic bacteria, very special in their requirement for respiration. They grow best in high concentration of carbon dioxide. Then it's present in atmosphere. Atmosphere is 4%, and they need approximately 10. The example of capnatic bacteria is Brucella. It's important animal and human pathogen, and it's a potential agent of a biological weapon because the bacteria cause severe chronic disease involving many organs in the human body. And why we paid so much attention for respiration of bacteria? It's necessary to provide them favorable condition in laboratory. It's no difficulties in cultivation of aerobic or facultative anaerobic bacteria. We just make inoculation at the surface of nutrition media and petri dish, and they produce the colonies. Uh, if we make inoculation of microorganisms in the liquid nutrition media, the aerobic bacteria grow uh, at the surface, producing the foil, because the concentration of oxygen is higher. Microaerophilic bacteria grow in some distance from the top, where the concentration of oxygen is lower, 5 to 10%. Facultative anaerobes can grow in all volume of this nutrition media because the concentration of oxygen is not essential for them. Aerotolerant bacteria mostly grow in the near the bottom of the test tube, and um, anaerobic bacteria produce the sediment. But really, obligate anaerobes cannot be grown in this condition just in nutrition broths. For them, we need special device named anaerostat with the chemical absorbent of oxygen. Only this condition without oxygen, the anaerobes can grow. And if anaerobes are very sensitive for oxygen, like Bacteroides or Fusobacteria, we need this anaerobic box to work with them. In this area, the air is replaced with inert gas. It may be N2 or CO2 or argon, and only in this atmosphere we can work with this very sensitive bacteria. All metabolic processes of biosynthesis or of respiration in microorganisms is possible only in participation of biological catalysts named enzymes. All enzymes, you know, it's proteins, and all live cells produce them, and enzymes very specific in their action. Um, each one catalyze only one biochemical reaction, and they very important in all metabolic processes of microbial cell. And here you see the classification of enzymes. According to their location, they may be exoenzymes, then produced outside the bacterial cell, and they active only outside. Mostly it's the hydrolytic enzymes, which necessary to cleave the large biopolymers before bacteria absorb their nutrients. And other enzymes are present inside bacterial cell, and they're named endoenzymes. Mostly the enzymes of biosynthesis and enzymes of respiratory chain, synthesis of ATP, they're located inside bacteria, the endoenzymes. According to their concentration, enzymes also divided into two groups: constitutive enzymes, their concentration mm, doesn't depend on condition of cultivation. It's constant. The example of constitutive enzyme like DNA polymerase, the concentration is constant in all occasions. And other enzymes, adaptive, bacteria produce this enzyme only if some substrate present in environment. In this case, bacteria produce more high concentration of this enzyme. Bacterial cell is very small, so it produces only necessary enzymes for adaptation, um, for nutrition media, for environmental conditions. And what is the practical importance of bacterial enzymes? I mean, in medicine. Many enzymes of bacteria act as effectors of pathogenicity. They can cause harm human body and affect the host cell. And here you see the list of this enzyme of pathogenicity, the most important of them. The hyaluronidase, which damages the connective tissue and help bacteria to spread in human body. Lecithinase, it destroys the cellular membrane and causes necrosis of cell. And we can detect this enzyme making inoculation of bacteria into blood agar. And it causes lysis of cellular membrane, including membranes of erythrocytes. And we see hemolysis around the colonies of bacteria if they produce this enzyme lecithinase. DNA that damages the nucleus of human cells and it's factor of necrosis. Plasma coagulase uh, produce a thrombosis in small blood vessels and it is interfere normal blood circulation, so it's very important in pathogenesis and many diseases. In neuraminidase enzyme, it cleaves neuraminic acid from the epithelial cell and it helps bacteria to attach to the human cell. And the attachment is the first stage of infection process. If bacteria doesn't attach, they easily removed from the mucosa, but after attachment, they start to multiply and colonize the surface and they invade tissue. So this enzyme very important in pathologic process causing this bacteria. And other reason why we started the bacterial enzyme that we can use them for identification of bacteria. Different bacteria have different enzymes. Each species of bacteria have specific set of enzymes. And we can detect the presence of these enzymes in special nutrition media to identify the microorganisms. Mostly we detect the saccharolytic and proteolytic enzymes. For saccharolytic activity, we use the nutrition media with carbohydrates and indicator. If bacteria ferment some carbohydrate, it produces acid products and the color of indicator is changed and we see that bacteria have this enzyme. For proteolytic enzymes, we use the nutrition media with gelatin and if bacteria produce proteases, proteolytic enzymes, it liquefies the gelatin and produce some gases like H2S, indole, and ammonia, and we can detect them with special indicator papers. And at this slide, you see the young microbiologist who produce the biochemical identification of bacteria using this his media. In each test tube, there is a one carbohydrate with indicator. And for biochemical identification, we make inoculation with one tested bacteria into each this test tube. And next day, we see if the color of indicator change or not. And then we compare these biochemical properties with properties of known bacteria, and in this way, we can identify the isolated culture. And at the table, this photo, you see other nutrition media to identify the microorganisms, the endomedium containing lactose and the indicator. If bacteria lactose positive, if they can ferment lactose, they change the color of nutrition media into pink. If they're lactose negative, no fermentation, the medium stay colorless. And it's very important in diagnosis of intestinal infections. And what is the aim of bacterial metabolism? Of course, it's bacterial growth and reproduction. Bacteria reproduce themselves only asexually by binary fission. And if we say microbial growth is doesn't about it's not about the growth of one bacterial cell. It means the increase of population of microbes. And the result of microbial growth is the colonies, the aggregation of bacterial cell, which we can absorb in solid nutrition medium. And here you see the schematic diagram of binary fission. You see that it's completely different from process of mitosis or meiosis, which characteristic for eukaryotic cell. Firstly, the bacterial chromosome is replicated and then bacteria grow. We see the division and as a result, the two identical cells produced. identical to mother cell. One cell divided into two, in next generation four, and in each generation we see doubling the number of cells. And bacteria multiply very fast if we provide them with favorable condition. They may divide each every 20 minutes, new generation appear, and in 7 hours, one bacterial cell may produce a billion live bacterial cells, multiply very fast. And when we cultivate the microorganism in nutrition media and measure the number of live microbial cells, we obtain this growth curve, which shows the dynamic of growth of bacterial culture, and it divided into four phases. The first phase, named lag phase, is the phase of bacterial adaptation. You see, the number of bacteria is stable. In this time, bacteria produce adaptive enzymes for new nutrition media. The second phase, the phase of logarithmic growth, exponential growth. In favorable condition, bacteria multiply with binary fission, produce new and new generation. And then next phase um, occur at the stationary phase. Um, you see the constant number of bacteria in some time, but there's a dynamic balance between new cells and cell death. Uh, in this phase, the concentration of nutrients decrease, so bacteria cannot multiply so fast like in previous phase. And the last phase, the phase of decline, of phase of cell death, due to accumulation of metabolic products, which may be toxic for cells, and they suppress their growth and cause the microbial death. But not all cells die. Um, some of them stay alive, and they take nutrients from dead cells or cells which undergo the autolysis. And these phases change one to another rather fast. All of them takes 24 hours. And the bacteria mostly metabolically active at the end of log phase and the beginning of stationary phase. And for scientific research, we need the fresh culture in this phase of their growth um, for, for biochemical studies. You know that we can observe single bacteria only under microscope, but colonies of bacteria visible with naked eyes in the nutrition media, and they have some shape, size, consistency, the pigmentation, and it names the cultural properties of bacteria, and we also use them for identification of microorganisms. Sometimes the appearance of colonies is very characteristic, like for these actinomycetes. For cultivation of bacteria in microbiological laboratory, we use the different nutrition media, and here you see the classification of media in different parameters. Mhm. And the more details about the most important groups of nutrition media. Mhm. In microbiology, we use ordinary or simple media. It's only two media in this group. It's the mid-peptone agar and mid-peptone broth, the liquid medium and solid medium. And they used only for prototrophic bacteria. The special media, the media for auxotrophic bacteria, with added some special compounds like serum or blood, potato starch, or animal tissues. And in this media, bacteria with special requirement can grow. Elective media produce a favorable condition only for one type of microorganism and suppress the growth of others. The example is the alkali peptone water, which is used for isolation of Vibrio cholerae. This bacteria can survive in high pH 9, and other bacteria which also can be present in intestinal tract of people prefer the neutral pH 7. And so in this media, they cannot multiply and we can isolate the vibrios. Enriched media used for primary inoculation of clinical specimen if the number of pathogenic bacteria is low, and we increase the number of these bacteria. And here you see the example of some media with indicators which help us to differentiate bacteria one from another. Mm. And for differentiation of bacteria, we used differential diagnostic media and for proteolytic activity and saccharolytic, hemolytic, and other types of biochemical activity. And they contain some substrates and indicators and help us to differentiate bacteria. Like here, you see the different species of microorganisms like E. coli, Klebsiella, Proteus, Enterococci, Staphylococci, two different species, produce the growth with different colors due to different biochemical activity. And it's very easy to differentiate them one from another only in one inoculation special nutrition media. And students, if you have any question, you may address them in the practical classes. We have a practical classes about physiology of microorganisms. And thank you for your participation in this lecture. And goodbye.